Hi, my name is Drew Roberts, and I'd like to thank you for just giving me your attention. Tough acts to follow, but I'll try and make it interesting for you. So, currently there's a contentious debate over GMOs, and normally it centers around what we consume, corn, things like that. But more recently, we've gotten into this area where we can create bacterially, plastic, things like that with genetically modified organisms. And specifically, Florida's come into the picture, or at least in the United States, of Key West. They have this Aedes aegypti mosquito who's, due to climate change, is coming up further and further from tropical areas into the subtropical areas, and it carries dengue fever and some other tropical diseases. And it's a pretty terrible disease. It could be fatal, but mostly it's just extremely painful, and all the economic resources you waste to treat people with this, they think, well, let's just control the population instead. And in fact, in Brazil and places all around the world, they've started doing this, but in the United States, we're still a little bit apprehensive. Anyways, the reason I want to look at this is just what might happen with population numbers. What can we do to target a vector species like this, and see what we can do about bringing it down without affecting other animals in the same, I guess you would say, ecological niche. And as far as this goes, we had success eradicating this organism before with DDT, but as we know, that also eradicated bald eagles. And so that's a negative side effect of that. So anyways, moving on, I had to begin my research by figuring out a way, how do I test this? How can I simulate, what might happen? My professor, Dr. Pearl, who helped me with this, gave me a great idea of, why don't you modify a Lotka-Volterra model to do this? And so we took a predator-prey model and kind of tweaked it a little bit. So it's not perfect, but it's a rough estimate. It's just an exploratory kind of research just to figure out what we might be doing. And so a couple of things I had to look at was like reproductive rate and mating habits of the mosquito, which it's pretty boring and it's tough to find information on. And it's actually really hard to test for. Mosquitoes it's a pretty ambiguous organism, and it's tough to find, it's tough to trap, they're very small. Anyways, I was able to sift through a lot of information. Most of it comes out of Brazil because they have an extreme problem with this. So, so now the reproductive rate is pretty good. I found a lot of ranges, so I decided to go with the lower end. The lower end seems to be more the norm. Rio de Janeiro is pretty much the same latitude as Key West, and temperature seems to have a huge effect on the reproductive rate, so I took the reproductive rate from Rio de Janeiro and just applied it to Key West. And so that's about 0.636. It's pretty good for an organism, and that kind of actually plays into how we want to use these GMOs. These GMOs, what they do is in the lab, they have a mosquito, males, are not the ones who bite, it's only the females who can transmit the disease. So the males, what they do is they take them in the laboratory and they put E. coli into the genome of the mosquito. And the only way that these mosquitoes can survive is if they're fed doses of tetracycline in the lab. So in the wild, when the gene gets transferred to the progeny, there's no way it can survive because it's not being fed tetracycline. So effectively, any female mosquito that's impregnated by a GMO male will not be able to reproduce. And the nice thing about this is they have such short lifespans that, there's a lot of ranges depending on tons of ambient factors, but it's about 13 days. So they only get one chance to reproduce. So if you can effectively remove that, you can do serious damage to the population. And so taking that on, and then we also have to look at how many times do they mate. And this is where a lot of the research came in, is the behavioral factors. And like I said before, it's pretty ambiguous. They're not quite sure. So I had to kind of estimate on this. Some say they can mate up to 3 times, but I just kept it at 1 because that was more of a typical baseline of what I found. It's possible they say a male can mate with a female up to 5 times, but that's if they get lucky. But and then another important aspect of that is that there is no real selective pattern in their mating. Their lifespans are short. It's dangerous out there for a mosquito, and they have only one chance. They're not selective, so there's no way that they can take any like polymorphic signal to kind of select. They just have to mate and hope for the best. So there's no kind of way that this will disrupt their mating pattern. They won't be able to smell, they're sickly or anything like that. So moving on to the more meat of the project. So putting this into the Lotka Volterra model, and then the population was the big factor. How big is the population? Well, estimates, once again, all over the table. It's like, it ranges from anywhere like about 110 per hectare, all the way up to thousands. They've recorded in certain like areas in India where sanitation conditions aren't properly maintained. Anyway, I use the lower end of the spectrum because Key West is a pretty properly maintained area. It's nicer, more affluent. Anyway, so Key West is about 7.4 square miles, which ends up being, pretty big area in terms of hectare. So it puts the population at about 220,000. And this is, again, the problem with the exploratory research is that the numbers aren't exactly hard, but it just gives us an idea. And so if you release about 1,000 males, is what I put the baseline at, what I tested at. You could release any number of GMO males, but the problem with that is you're going to have to keep applying it. And that's the real variable here, is what we control, is how many and how often you release. And so if you release 1,000 males every 4 generations, it'll take about 37 generations to put the population from 220,000 all the way down to below 40,000. And then it's, after that, you get isolation of genes. Once you reduce that kind of genetic variability, the organism's not as viable. Bad genes get isolated in the population, and it just tanks after that. And then every 10 generations, which is kind of longer, it's after 70 generations you see a drop below 40,000. And then if you apply it every generation, it only takes 13 generations for it to go down into this level where it's not going to rebound from. So, and like I said before, it's about 2 weeks per generation. So within a matter of a year or two, you can completely eradicate this organism from an area and remove the danger of dengue fever, which is actually pretty prevalent in the Key West area. It's pretty high rate of contraction compared around. And so basically, what I'm trying to get at is keep an open mind about this and just look at what the data shows. And really, the only claim to be made is that it's a highly effective method compared to pesticides, which most insects develop resistance to. They're effective at pretty low rates, actually, and you have to find the nests. But these guys, you can just release them wherever, and eventually the population will decline to an area, or decline in an area to where it's not dangerous to the population. Pretty much it. Thanks, guys.